don't like low oxygen conditions, and they only like oxygenated conditions, but they didn't seem to have the same problems in my site. I'm not entirely sure why. Anyway, if you were to pull everything together and go through my section of core from the earliest point in time to the present, we start with marine isotope stage 6. We've got an abyssal to a lower bathyal setting, high sedimentation rates, high productivity, low oxygen, because what happened was there was a sea level regression, and it caused the currents, the bottom water currents operating in that area to eddy, and so you had less oxygen. Marine isotope stage 5, it was the same marine setting, but a transgression allowed more oligotrophic conditions to return to that region of the Tasman Sea. There was an early temperature maximum, which caused a jump in sedimentation and sea surface productivity with only a slight drop in oxygen levels. This period typically had high winds, low sedimentation rates, relatively low temperatures, and relatively high sea surface productivity. Marine isotope stage 4 is again the same marine setting. It was a comparatively warmer glacial period, had low sedimentation rates and oxygen levels. Excuse me, I'm still burping with beer. So, marine isotope stage 6 is again still the same marine setting. You have oxygen levels similar to the previous stage, but like marine isotope stage 5, it was characterized by high winds, low temps, relatively high productivity, despite low sedimentation rates. Now, when things get interesting, in marine isotope stage 2, we've changed to an upper bathyal to continental shelf. There was a considerable sea regression which caused abyssal taxa to become stressed out. So foramen assemblage 1 got stressed, oxygen levels dropped, and they either have very, very low abundances or they completely died out. The lower oxygen levels fluctuated throughout, and they provided a lot of environmental stress, but they allowed those infaunal taxa to flourish because they really liked that kind of setting. So then in marine isotope stage 1, we have a slight increase in water depth again to a lower bathyal to a continental shelf. So the transgression between marine isotope stages 2 and 1 coincided with more arid surface conditions. The sedimentation rates remained the same, but oxygen levels increased quite a bit. And this increase in oxygen caused foramen assemblage 2, which loves low oxygen conditions, to also get stressed and die out. Well, not die out completely, but they certainly reduce in numbers. So after sea level rise stopped at around 6,000 years BP, the dominant foraminiferal abundances in this region are comparatively low to previous occurrences. All right, so for this study, everything that I can pull together is basically that relative abundance, biodiversity, and distribution of the most abundant foraminiferal taxa revealed 2 assemblages. Foramen assemblage 1 was dominated by opportunistic epibenthic taxa that thrived in abyssal to bathyal marine settings with high oxygen levels from marine isotope stages 6 through 3. Foramen assemblage 2 was dominated by infaunal continental shelf species adapted to low oxygen conditions throughout glacial stage 2 and wane to the present. Disaerobic conditions caused species dominance to shift at various horizons identified by key taxa. And the presence of deep oceanic current indicator taxa and their relatively consistent frequency throughout this section of core has also brought home that the deep sea oceanic current circulation in this region hasn't changed a lot in the past 140,000 years. So, moving right along. So the natural hazard work was sort of part of my PhD, and basically the idea was, could you fingerprint tsunami deposits and also potentially extreme storm deposits using foraminifera? One of the key questions in paleo natural hazards research is, can you tell the difference between a paleo tsunami and a hurricane or a cyclone or a big storm event? And also, basically, so this work was after the Indian Ocean tsunami, but before Solomon Islands and before Japan and before a whole pile of other stuff. And so the idea was, after the 2004 event, we wanted to gain as much information as possible on tsunami to get a very confident paleo-tsunami and paleo-extreme event history of a region to see how much danger we might potentially be in. So we had some funding from the Australian government to figure out whether we needed to worry about tsunami or not. And so, because there was all this funding getting involved in tsunami research, particularly sedimentologists were increasingly looking for a broad array of techniques that could enhance what they were finding, basically get a more confident assessment of the hazard history of a region. So first off, I started off with a review. I looked at every single paper up to that point that had used forams in tsunami assemblages, and these are all the locations up to about 2008 that people had bothered with forams in tsunami deposits. There was very little consistency in what was found in relating to those recovered assemblages and the types of data. So some people, practically no one, identified the foraminifera to species level, which is fair enough because a lot of the people were tsunami sedimentologists. They weren't taxonomists, they weren't micropaleontologists. So lots of people just said, tsunami deposit, it is different because it has forams and above and below doesn't. Or they said things like, we have marsh forams above and below, and here so-and-so told me that these are marine shelf forams. Ta-da! So there was, they weren't used in really that much detail. But what certainly came clear as I was reading more and more of these papers, that because the locations that tsunami come into land is all over the world in all these differing localities with different topography, different bathymetry, different sediments, the deposits that you're finding are just as varied as the localities that you're looking at. So, as I said, there was a broad range in the sorts of data that I was getting. There was no way that from doing a literature review I could say, well, this is what you're going to have with forams in a tsunami deposit, because they were found in a wide range of different sediment types. There was a tendency to note the transport of open water sort of sediments to a completely different marine, completely different depositional environment. So people only want to talk about the types of forams in the tsunami deposit if there was a really obvious difference. So as I mentioned, if there was an open marine or a continental shelf assemblage that was dumped in a marine marsh, not a marine marsh, in a brackish marsh or sort of lagoon area, because the foram assemblages were so extremely different, they were happy to talk about that, but they weren't happy to mention the types of forams found if it was, say, from only further up the coastline, or it was only from a 30-metre depth brought up to a 2-metre sort of water depth assemblage. So there was that sort of tendency. The size and shape of foraminifera could influence how much sorting and breakage, and also whether they would be picked up by the tsunami or not. So there was some deep sea foram work that was done off the east coast of Japan that found that there was this particular foram species that kind of was shaped like a flying saucer, and that made it far more likely to be picked up because it was more fluid, had better fluid dynamics. So it would be more likely to be picked up than other forams and transported by the tsunami. And then I had some people telling me that the tsunami picked up the sediment and dumped it and then went away. So because there was only one event, the forams were preserved perfectly. But then I had another event say that because the forams were picked up and moved back and forth by the series of waves associated with the tsunami, that the taphonomy was terrible and they were very broken and damaged. So there was this massive, contrasting data with these different tsunami events. So what I did find that was, where possible, depending on the situation, depending on the locality, and depending on how much effort people wanted to go to with their involvement of foraminifera in tsunami deposits, you could find out, using forams, the depth of water from which the sediments were entrained. So, for instance, there's this great paleo-tsunami deposit of the Boso Peninsula on the east coast of Japan, and they've found forams from a 90-metre water depth that have been picked up and dumped. So it can give you a real good idea of where the tsunamis come from and what sort of depths that wave base was reaching. You can get the distance of sediment transport, you can get the hydrodynamics of the tsunami, flow velocity, turbidity, and post-depositional environmental processes. You can look at what's happened since deposition by looking at the condition of the forams. The best application of forams incorporates the identification of the species and gleaning any information you can from what those species can tell you, looking at taphonomy, and then aligning this data with other proxies and with other types of information investigation to bring this big whole picture sort of study together. And that was exemplified by Andrea Hawkes in 2007, looking at the Indian Ocean event. She looked at forams, diatoms, mollusks, loss on ignition. She did dating. She did everything she possibly could, and she got a very, very clear picture of her tsunami deposit. It was really, really well done. All right, so I was involved with two particular studies. I wasn't the principal investigator in either one, but the first one was looking at this site, an unusual coarse, shelly sedimentary unit a few hours south of Sydney. So basically this is, Sydney's not on here, this is Wollongong. Wollongong is about 2 hours drive south of Sydney, and we're looking at Bateman's Bay down here. This is Bateman's Bay. So what basically happened was we did samples from inside Bateman's Bay and then up here, right up this channel behind a bridge and this big sandbank was where we found this really weird deposit. And so obviously we thought that it would be from high energy depositional event. And we looked at sedimentology, macro, micro, paleo, radiocarbon, and amino acid racemization. So this is the deposit here. You can see it pretty clearly. This is, I was only maybe 20 centimetres above water level when I took this picture. So it's about a metre and a couple of metres, 2 metres above water level. You can see it's full of these really big, coarse seashells. This is, I think, Anodara trapezia, I think. I'm not very good with mollusks. Anyway, so we used them for dating, and we had two lots of dating because we had one dating set that said those shells were dated from between 1,000 and 1,300 years BP, and we had another set of dating that said they were between 2,000, they were around 2,500 years BP. Now, there's very little historical data on events within Australia because we're a relatively young country in regards to written records, and so there was no record of a tsunami event around the sort of 1,000, 1,300 year, but there was a 2,900 BP tsunami event. So it could potentially be related to that. Well, we're not really sure. So basically I found a whole pile of forams, but in the units above and below, the forams were either at lower abundances or they were absent. And there was a difference in the taphonomic grade. However, these forams weren't that different from the foram assemblages that I was finding in Bateman's Bay. They actually looked like they were just forams from the surrounding area. They weren't brought in from a big distance. And so because of the dating and because we didn't have a really clear signature, we couldn't conclusively say that it was a tsunamigenic deposit. Though I have noticed that there's a paper that's about to come out on the Australian tsunami history, and they think that this is, so they've re-looked at our data, and they think that it was a tsunami deposit associated with a 2,900 BP event. Anyway, so the next project I was involved in was looking at a cyclone deposit, or a hurricane deposit, I think is what you call them up here. So basically I was doing work on the Great Barrier Reef. I was going there every year anyway, and then in March in 2009 there was a Category 4 cyclone event that went right over Heron Island, so well, almost all over Heron Island. This is the path of the cyclone, and here's Heron Island, and this is what it looked like from above. And so I was able to sample before, because I've been going there regularly anyway, I sampled before the event, within a week after the event, and then because I was still going back there, I sampled 1 year post and 2 years post the event to see if you could see a recovery in the assemblage. So we basically applied, obviously we did identification, we did counts, we applied Shannon-Wiener, Bray-Curtis, alpha, Paley's evenness. We threw everything we could at it. And so these are the dominant foraminiferal species that I found. This is Fisher alpha, Shannon-Wiener, and Paley's evenness. So the blue is 2008, red 2009, 2010. So this is pre, and these two are post. And as you can see, while there's not a lot that is positive about the difference between the three. Sometimes the pre-cyclone event is richer, sometimes it's not. And so there wasn't a nice clear signature. So then we looked at the Curtis Cumo cluster, And again, we weren't getting that much that looked very good. And then we thought maybe if we took out the larger benthic foraminifera and just looked at the smaller heterotrophic taxa, which might be a bit more sensitive to big events, maybe they might show us something, but no. No, nothing really. And then we looked at grain size, and then there wasn't a real big difference in grain size either. So these are the grain size fractions over pre and post. This is a little bit finer, but it's nothing significant. It wasn't telling us anything. And then we looked at taphonomic grading. So I think Yordanova and Hohenegger did some work off Okinawa and Seisoku Island in the south of Japan, and they assigned a taphonomic grade. And so we applied that taphonomic grading system to see maybe if there was a taphonomic difference between pre and post, but not really. So again, this is the total assemblage. Blue is pre and green and red is post. This is just the larger benthic foraminifera, and this is just the smaller benthic foraminifera, and there really is no difference between the two. So finally, we came to canonical correspondence analysis, and lo, this is the pre, and this is the post. Hooray! but this is pre, this is 2009, so straight after the event. This is 2010, and this is 2011. So if we had a pre-assemblage and then we had a post-assemblage and then you were going back to the pre-assemblage, so you wanted to recover, you would be expecting it to go this way, or even in between here. It should be like 2009, 10, 11, or 2009, 10, 11, but it's not. It's still, yeah. So, despite evaluation of data being complete, there is no significant change in population structure, abundance, diversity, or taphonomic degradation across these sampled phases. But this attests to the tenacity and resilience of reef-flat foraminiferal assemblages to extreme storm events in cyclone-prone regions. So it highlights the possible danger of drawing conclusions about possible paleo storm events or cyclone events based on one single faunal proxy. And it highlights that multiple proxy approaches supported by statistical analyses are essential before the status of an extreme event deposit can be validated in the rock record. All right, so conclusions for forams and natural hazards. Their use is still being developed, well and truly, but we're getting better. I promise. Their potential effectiveness is greatly dependent on the dynamics of their depositional setting. Hazard depositional settings and their deposits are incredibly diverse, and therefore a diverse suite of techniques need to be applied to accommodate for this variance. And then where appropriate, foraminifera have great potential to provide meaningful information regarding the nature of various natural hazards within a region. Okay, so last part. I think I'm going okay if I go through this relatively quickly. There's lots of pretty pictures in this, so I thought it might be nice to end. So I did a baseline ecological study, so I wanted to provide a basis of comparison for studies on the Great Barrier Reef, the Southern Great Barrier Reef, and figure out what features determined foram distribution in that area. So, and it was a pretty sweet place to do some field work. So this is the Great Barrier Reef when you put it on one nice big long line. These are all places where people have studied forams on the Great Barrier Reef, and we're looking at this little section down here. So there tends to be a lot of work in the central GBR. So these are the 4 reefs I studied, with Starry Reef, Heron Island, and Sykes Reef. These 2 are actually joined by an old cast platform, and then we have One Tree Reef down the bottom. So I looked at previous work because there was, as I said, there was lots of work already established on the GBR. So Sven Uthik found across 27 reefs that heterotrophic species flourished in high organic content and low light availability. Uthik and Nobes across 20 reefs found assemblages influenced by substrate type, current wave energy, temperature and depth. Melissa Lobagaya looked at low isles, and she said reef zonation, substrate and architecture of the reef flat determined foram assemblage. Backart looked at lizard reef, Palfrey and South Islands, and he said algal cover was the most important thing for sustaining alive communities. And John Gell looked at Heron Reef and said the highest concentration of living forams was within the protected lagoonal waters of the reef. So I did 4 transects on Heron Reef because that had a really great reef flat to work on, the dynamics of well, I couldn't walk on Wistaria Sykes Reef, and then One Tree has a completely different sort of shape, so I couldn't do transects along those other reefs. But I did use a little dredge thing to get a nice clump of sediment. Lots of samples. I picked 30,000 forams, and it's quite a few, especially to identify them. I did stain them for 24 hours. I looked at macroalgal presence, absence, and then I used the same suite that I'd been relying on for a lot of my analyses, and I also looked at grain size. So what I found was Calcarina hispida, this dude, was 39% of my total forams, and it was particularly found in depths of less than 2 metres. Bacilogipsinus ferrulata, 10.5%, that was the number 2 contender. Similar distribution to Calcarina, more dominant in the rampart and rubble zone of the reef. Quincaloculina neostriatula was 5% of my totals and was most common in depths of between 2 and 7 metres. And then Amphistogina species were abundant, but they were particularly abundant in my 1 channel sample, and they accounted for 29% of that sample, and that was taken from a depth of about 35 metres. So if you want to look at what that's like on a reef, this is Heron Reef, that's Sykes and One Tree in the background. So this is where Calcarina hispida was really happy. Around here was where Bacilogipsina was really happy. In this sort of big patch reef lagoon was where we had Quincaloculina, and Amphistogina was super happy down here somewhere. So I looked at, as I said, I looked at live assemblages, but I didn't look at, I didn't collect algae for forams, I just collected sediment. So I only had 17 species of living forams from 56 of my sites. So the most common was Miletiana milleti and Cymboleperetta bradii. And they had, the main distribution was between a mixture of reef flat and lagoonal sites. What was really common, the most common way for me to find living forams was when they were juveniles, and they were always stuck to bits of reef rubble and other forams. So this is a Rotaliamina chitinosa, and it's attached to a giant Marginoptera. So yeah, it was only because they were studied in bright pink that I could even find them. So this little guy's ester stuff in there, and then this is Miletiana milleti on a piece of coral rubble, and yeah, they're pretty small. Right. So I did look at the algae, the most common. There was 25 algal species, the most common being the turtle grass, along with Amphiroacrassa and Turbinaria ornata. So these were the 4 transects I did. This is Heron Island, and this is the reef flat. So on this transect, I found that there was sediment coverage, and this is sediment coverage, and this is algal, the number of algal species, and this is the abundance of those 2 most dominant taxa. So you can see that as you head out to the reef edge, sediment coverage obviously decreases because you're getting increased coral. So is your algal diversity, and the foram counts. Calcarina hispida was the most happy in the inner reef flat, and Bacilogipsina was actually happy further out on the reef edge. And then when you looked at transect 1, which is along the inner and outer reef margin, you had a small variance across that zone, but where algal diversity was lowest, sediment coverage and Calcarina